In this module, we'll look at the differences between RNA and DNA. We'll also look at some of the chemical properties of these molecules. First of all, let's look at the differences. As I have mentioned before, RNA has ribose sugar and it has uracil. However, DNA has deoxyribose and thymine instead of uracil. Now let's look at some of the chemical properties of these molecules. As I've mentioned in previous module that these molecules are polymers which are held together by phosphodiester bond. The important aspect of the polymer is that two ends of this polymer are different. One end is called the 3' end and the other one is called the 5' end. They have different properties and I will highlight those. Here, for example, we have an RNA molecule. It has 3' end over here because the 3' end basically means the carbon atom which has a free hydroxyl group. Here we have the 5' end in this RNA molecule. This carbon here is the 5' carbon. It is attached to a phosphate group. So this difference in two ends of this nucleic acid are important because when this polymer is elongating or new monomers are being added to this polymer, they always add on the 5' end and not at the 3' end. Also, you will notice that these two ends, they're different in this RNA molecule here. And if we draw a line between 5' end and 3' end and put an arrow on it, we basically get some sort of polarity in this molecule. It is important to note this here because when we go to DNA, DNA is not single-stranded. It has two strands. RNA is single-stranded, which we have seen here. The two DNA strands run anti-parallel. What do I mean by that? If one strand is in direction 5' to 3' direction, the other one will be 3' to 5' direction. So these in DNA we have two strands rather than one strand and they're running anti-parallel. In RNA we generally have a single strand. So these are two important aspects of these two molecules, important differences also. Now let's look at some other properties of these molecules. DNA and RNA, they contain information as a result of the sequence of those nucleotides. We talked about the nucleotides A, G, T and C, or U in case of RNA. This information in DNA is present inside. It is not present on the outside. Like in proteins, the three-dimensional structure is important. DNA does not have a varying three-dimensional structure. In fact, it is uniform. It has a uniform diameter. The information this molecule carries is the sequence of the nucleotides, not the three-dimensional shape as was the case in case of proteins. So now we have a dilemma that we know that DNA is carries the genetic information. However, we cannot use this genetic information without proteins, and DNA cannot replicate without proteins. We'll talk about this process in more detail when we talk about DNA replication. But the dilemma here is what came first in evolution. If the DNA came first, it would not be able to replicate itself, or it will not be able to do any functional work without the presence of proteins. Proteins, on the other hand, need something to code for them, and DNA codes for proteins. So what came first? What we are talking about is that we need two types of activities in order to sustain a system, a living system: genetic information and enzymatic activity. There is another molecule that can perform both these functions. That molecule is the RNA molecule. RNA molecule, as we saw, is a single-stranded entity. It can fold on itself and it can form complex three-dimensional structures. Also, it is made up of nucleotide polymers. Nucleotides, it's a polymer of nucleotides. So it can code genetic information and has enzymatic activity. The next question becomes that if RNA is such a marvelous molecule, it can have both these activities, coding for information, genetic information, and enzymatic activity. Why don't higher organisms have RNA as genetic material rather than DNA? The answer is the hydroxyl group, additional hydroxyl group in RNA, pentose, makes it more prone to degradation. DNA is relatively more stable than RNA. That is why DNA was the preferred choice during the course of evolution as a molecule which would carry hereditary information.